Chapter 6, Microbial Nutrition and Growth. So I'm going to cover the first topic, growth requirements, along with this chapter overview. Then we're going to hit culturing microorganisms and growth of microbial populations. So here's the first topic in Chapter 6, growth requirements. So the learning objectives for this topic are: describe the roles of carbon, hydrogen, oxygen, nitrogen, trace elements, and vitamins in microbial growth and reproduction. Compare the four basic categories of organisms based on their carbon and energy sources. Distinguish among anaerobes, aerobes, aerotolerant anaerobes, facultative anaerobes, and microaerophiles. Explain how oxygen can be fatal to organisms by discussing singlet oxygen, superoxide radicals, peroxide ion, hydroxyl radical, and describe how organisms protect themselves from toxic forms of oxygen. Define nitrogen fixation and explain its importance. Explain how organisms have physical requirements for growth, including specific conditions of temperature, pH, osmolarity, and pressure. Describe how quorum sensing can lead to the formation of a biofilm. So for this topic, we're going to be talking about nutrients, chemical and energy requirements, and physical requirements. So under nutrients, because this is such a big topic, we're going to be cutting it down into subtopics. Actually, there's subtopics. So we're going to talk about the sources of carbon, energy, and electrons that microbes need, oxygen requirements, nitrogen requirements, and other chemical requirements. First, let's talk about what we mean by growth requirements. Remember, with microbes, growth equals reproduction as well as an increase in size. What can we measure easily? Reproduction. An increase in the number of cells in a population. All organisms need three things to grow, to get bigger, and to reproduce. They need an energy source, they need a carbon source for their organic molecules, and they need an electron source. So first we're going to talk about energy and carbon sources, because we often group these into four terms or four groups of organisms. Let's start with the photoautotrophs. So photo, the first part of this term, refers to where this type of organism gets its energy from, and that's from the sun. Auto, which means self, means where they get their carbon source from. So this doesn't mean that they get their carbon source from their own cells. It means that they're able to acquire the carbon for their carbon skeletons without eating something else. What does that mean? They are able to fix CO2. So from the last chapter, that would mean that photoautotrophs have both light-dependent reactions and light-independent or carbon fixation reactions. So it's easier for me to remember things if I have an example. So what's the most famous autotroph example that you can think of? That's right, plants. When we're talking about microbes, the example I want you to remember are cyanobacteria. They get their energy from the sun and they fix CO2, and they're also closely related to chloroplasts in plants. Next, chemoautotrophs. Now these are weird guys. They're able to, well, let's go in order. So they get their energy from chemical sources. So they get them from chemicals. The auto means that they're able to fix CO2. So these organisms do not have a light-dependent reaction set up, but they do have the enzymes to be able to fix CO2. So they have to get energy from chemicals. So then there are the reverse of these guys. They get their energy from the sun, so they have light-dependent reactions, so they can get energy from the sun, but they can't get their carbon for their carbon skeletons, for their organic molecules, from CO2. So they have to get them from chemicals. Now for the last one, chemoheterotrophs. You probably will be able to tell the breakdown of this now. Chemo means they get their energy from chemicals. Hetero means it's from others, not able to fix CO2, so they get it from chemicals. You are chemoheterotrophs. The coli in your gut are chemoheterotrophs. So let's talk about these weird guys in the center. We have eukaryotic examples and prokaryotic examples of both photoautotrophs and chemoheterotrophs, but we only have prokaryotic examples of these two. So an example, a chemoautotroph, would be there are bacteria that live in the deep ocean on deep sea thermal vents. And so they are able to get their energy from the heat and from the chemicals that are coming out of those deep sea energy vents. They're able to fix CO2, so they don't have to get their carbon skeletons from another source, but they do have to get their energy from something other than the sun. Photoheterotrophs. There's a fun bacterium called Halobacterium salutarium. I think I said the species name right. They live in high salt concentrations. We often find them in evaporation pools, and they have purple bacterial chlorophyll. They use that energy to make ATP, but they do not have a carbon fixation pathway, so they have to eat other organisms to get their carbon molecules, to get their organic molecules. So these are the four categories you need to know: photoautotrophs, chemoautotrophs, photoheterotrophs, and chemoheterotrophs. Now let's talk about electron sources. Now, generally speaking, electron is so common that it is not what we call a limiting resource. What do I mean by limiting resource? If you have something that's rare, for example, nitrogen, which we will be talking about later, it limits the ability of an organism to grow by its concentration. So in other words, in the environment, you're going to have less usable nitrogen than you are hydrogen, so the organism is not limited by the concentration of hydrogen. Having said that, let's talk about where organisms get their high energy electrons from. Now, what do they use their high energy electrons for? That's right, redox reactions, and the king of all redox reactions, electron transport chains. These are generally part of hydrogen, and there's two different ways that an organism can get these high energy electrons. There are the organotrophs. You're an organotroph, by the way. And they get their electrons from the same organic molecules that provide carbon and energy. So if we were to name you, you would be a chemoheteroorganotroph. You thought the chemoheterotroph was long enough, huh? Now, we do have some funky organisms called lithotrophs. They get their electrons / hydrogens from inorganic sources. Some of the places where we find these organisms are on mineral deposits. So you're going along, you find an exposed iron ore site. You're probably going to find a lithotroph that are using the electrons from those iron molecules in the iron deposit for their high energy electrons. Kind of creepy, huh? So here are some of the inorganic sources that lithotrophs can get their electrons / hydrogens from. Now, just to keep things easy, unless we tell you something is a lithotroph, it's an organotroph. So on to oxygen requirements. Now, just so you know, some organisms are killed by oxygen. That seems kind of weird to us, because if we're cut off from oxygen, we die. And most of the organisms that we're able to grow in this lab, or the lab for this class, need oxygen. So how is oxygen toxic? Well, it's not toxic, but oxygen byproducts of redox reactions are. So there are 4 of them. We're going to go through them step by step, and we're also going to talk about how the cell defends itself from oxygen byproducts of redox reactions. So the first one is singlet oxygen, and we represent it like this. Well, what's happened is, during the redox reactions, some of the electrons of the singlet oxygen have been energized. And this happens during aerobic respiration and during exposure to light. So there's singlet oxygen in the atmosphere. And remember, the electrons are at a higher energy. They're ready to go to some other atom, or they can drop down, releasing the energy. In any case, singlet oxygen goes around reacting with macromolecules that it shouldn't. Some cells make use of this. Your white blood cells oxidize pathogens by producing singlet oxygen. So they engulf the pathogen through endocytosis, through phagocytosis, and then they expose the pathogens to singlet oxygen in high concentrations, and they die. How do your cells prevent themselves from being killed by singlet oxygen? Well, there are large molecules that are pigmented that we call carotenoids. Two guesses on where we first isolated carotenoids from. That's right, from carrots. And carotenoids have a tendency to be either orange or yellow. And these pigments, these carotenoids, are very good at taking care of singlet oxygen. And they move them down to superoxide radicals, which we'll be talking about next, obviously. So this is part of why they tell you to eat your orange and your yellow vegetables. So superoxide radicals are produced when carotenoids take care of singlet oxygen, and we represent them like this. They are formed during incomplete reduction of oxygen during electron transport. So when oxygen is being the last terminal electron acceptor, it picks up the electrons, but it hasn't picked up a hydrogen yet. So it wanders around, it's got a negative charge, it wanders around pulling hydrogens off of molecules that shouldn't have hydrogens pulled off of them. So some anaerobes, when they undergo metabolism in the presence of oxygen, will produce superoxide radicals, even if they do not have an electron transport chain, and it kills them. Because superoxide radicals are very reactive, very toxic, they go around pulling off hydrogens even more so than singlet oxygen. How do, say, your cells, which are producing superoxide radicals all the time, how do they protect themselves? How would, say, the E. coli in your gut, when it's being grown in the presence of oxygen in the lab, how does it survive superoxide radicals? Well, organisms that are aerobic produce enzymes called superoxide dimutases. And superoxide dimutases usually have a metal atom as a part of their active site. And what they do is they take the superoxide radical and they attach hydrogens to it, which is our next thing. We take 2 hydrogen ions and add it to our superoxide radical, and we get hydrogen peroxide. But hydrogen peroxide, or the peroxide anion, we show it like this, is not terribly good for cells either. So hydrogen peroxide, in fact, hydrogen peroxide has become a big active ingredient in surface disinfectants because they go around pulling hydrogens off of things. So superoxide dimutases and other metabolic reactions cause hydrogen peroxide. So it's highly reactive, it's an oxidant. Okay, it oxidizes molecules by removing electrons, by removing hydrogens. So here's how we protect ourselves against it. There's an enzyme called catalase. And what it does is it takes hydrogen peroxide and it breaks it down into water and oxygen. So if you ever had a cut and you've poured hydrogen peroxide on it and it bubbles, well, that's because your cells have catalase. You're forming water and oxygen, and the oxygen is bubbling off. Now, other organisms have peroxidases, but catalase is the one that I want you to remember.